The discovery of CRISPR-Cas9 has ignited a revolution in genetic engineering. This groundbreaking technology allows scientists to edit DNA with unprecedented precision, offering new hope for treating diseases and addressing global challenges. CRISPR, short for clustered, regularly interspaced short palindromic repeats, is a natural defense mechanism found in bacteria. Scientists have harnessed this bacterial system to create a programmable gene editing tool. The CRISPR system has rapidly become an indispensable tool in laboratories worldwide, accelerating research in fields like medicine, agriculture, and environmental science. Its relative simplicity, affordability, and versatility have democratized gene editing, making it accessible to a broader range of researchers. Scientists are developing CRISPR-based therapies for genetic diseases like cystic fibrosis and sickle cell anemia. In agriculture, CRISPR is being used to engineer crops that are more resistant to pests, diseases, and climate change. However, this revolutionary technology also raises profound ethical questions that must be carefully considered. CRISPR-Cas9 acts as a precise pair of molecular scissors, allowing scientists to cut and paste DNA sequences. The system consists of two key components: the Cas9 enzyme, which acts as the scissors, and a guide RNA molecule, which directs the scissors to the desired location on the DNA. The guide RNA is designed to match a specific DNA sequence, ensuring that the Cas9 enzyme cuts at the correct site. The process begins with the guide RNA binding to the target DNA sequence. This binding guides the Cas9 enzyme to the same location, where it makes a precise cut in the DNA strand. Once the DNA is cut, the cell's natural repair mechanisms kick in. Scientists can exploit these repair mechanisms to introduce changes to the DNA sequence. There are two main ways to repair a DNA break: non-homologous end joining NHEJ and homology-directed repair HDR. The simplicity and versatility of CRISPR-Cas9 have made it a game-changer in genetic engineering. CRISPR's potential to revolutionize medicine is vast. Scientists are exploring its use in treating a wide range of diseases, from genetic disorders to cancer. One promising area of research is using CRISPR to correct genetic defects that cause inherited diseases. CRISPR also holds promise for treating infectious diseases. In the fight against cancer, CRISPR is being investigated as a tool for engineering immune cells to recognize and destroy cancer cells more effectively. Beyond human health, CRISPR is transforming agriculture. Scientists are using CRISPR to develop crops that are more nutritious, resistant to pests and diseases, and tolerant to harsh environmental conditions. These advancements have the potential to enhance food security and sustainability in a changing climate. While CRISPR offers tremendous promise, its unprecedented power to alter the genetic makeup of organisms raises profound ethical questions. One major concern is the potential for unintended consequences. Altering one gene could have unforeseen effects on other genes or the organism as a whole. Thorough research and rigorous testing are crucial to minimize the risk of unintended outcomes. The possibility of germline editing, which involves making changes to DNA that can be passed down to future generations, raises particular ethical concerns. Some argue that germline editing could be used to eliminate inherited diseases, while others fear it could lead to the creation of designer babies with specific traits, exacerbating existing social inequalities. Access and equity are also major concerns. CRISPR technology is rapidly advancing, but access to CRISPR-based therapies and agricultural products may not be evenly distributed. Ensuring equitable access to these innovations is crucial to prevent further disparities in health and well-being. International guidelines and regulations are being developed to govern the use of CRISPR technology. The field of CRISPR research is rapidly evolving, with new breakthroughs and applications emerging regularly. One exciting development is the discovery of new CAs enzymes with different properties. For example, Cas13 enzymes target RNA instead of DNA, offering new possibilities for studying and treating diseases caused by RNA viruses. Cas12 enzymes, on the other hand, can make multiple cuts in DNA simultaneously, expanding the toolkit for complex gene editing. Base editing is another groundbreaking development in CRISPR technology. Traditional CRISPR-Cas9 makes a double-stranded break in DNA, which can lead to unintended genetic alterations. Base editing, in contrast, allows for the precise modification of individual DNA bases without cutting the DNA strand. Prime editing, a more recent advancement, takes precision gene editing to a new level. These breakthroughs highlight the rapid pace of innovation in the field of CRISPR. Beyond its transformative impact on medicine and agriculture, CRISPR is poised to revolutionize a wide range of fields. In environmental science, researchers are exploring CRISPR's potential for bioremediation, using engineered microbes to clean up pollutants and restore damaged ecosystems. CRISPR could also be used to develop novel biofuels and biomaterials, paving the way for a more sustainable future. In the realm of synthetic biology, CRISPR is enabling scientists to rewrite the genetic code, creating organisms with novel functions and capabilities. This field holds promise for developing new medicines, biomaterials, and even biocomputers. CRISPR is also transforming the field of diagnostics. Researchers are developing CRISPR-based diagnostic tools that can rapidly and accurately detect pathogens, genetic mutations, and other biomarkers of disease. From eradicating diseases to addressing climate change, CRISPR has the potential to shape a brighter future for humanity. Cancer, a complex group of diseases characterized by uncontrolled cell growth, remains a leading cause of death worldwide. Traditional cancer treatments, such as chemotherapy and radiation therapy, often come with debilitating side effects and limited efficacy. CRISPR-based therapies offer new hope for more precise and effective cancer treatments. One promising approach is using CRISPR to enhance the body's own immune system to fight cancer. CAR T-cell therapy involves extracting a patient's T-cells, genetically modifying them to recognize and target cancer cells, and then infusing them back into the patient. CRISPR can be used to improve the effectiveness of CAR T-cell therapy by making T-cells more effective at recognizing and destroying cancer cells. CRISPR can also be used to target and disable genes that are essential for cancer cell survival and growth. While CRISPR-based cancer therapies are still in their early stages of development, they hold immense promise for revolutionizing cancer treatment and improving patient outcomes. The global population is projected to reach 9.7 billion by 2050, placing increasing pressure on food production systems. Climate change, pests, and diseases pose significant threats to global food security. CRISPR technology offers powerful tools to address these challenges and enhance agricultural productivity. CRISPR can be used to develop crops that are more resistant to pests and diseases, reducing crop losses and the need for harmful pesticides. For example, researchers have used CRISPR to create wheat varieties resistant to powdery mildew, a fungal disease that can devastate wheat yields. Drought and salinity are major environmental stresses that limit crop productivity in many parts of the world. CRISPR can be used to engineer crops that are more tolerant to these harsh conditions, expanding agricultural production into marginal lands. By enhancing crop yields, resilience, and nutritional value, CRISPR has the potential to revolutionize agriculture and contribute to a more sustainable and food-secure future. The discovery of CRISPR-Cas9 has ushered in a new era of genetic engineering, offering unprecedented opportunities to understand and manipulate the building blocks of life. From treating diseases to addressing global challenges, the potential applications of CRISPR are vast and continue to expand as research progresses. CRISPR's impact on medicine is already being felt, with clinical trials underway for various genetic disorders and promising advancements in cancer treatment. In agriculture, CRISPR is enhancing crop yields, resilience, and nutritional content, contributing to a more sustainable and food-secure future. However, the immense power of CRISPR also raises profound ethical questions about unintended consequences, germline editing, and equitable access to these transformative technologies. Responsible innovation requires ongoing dialogue and collaboration among scientists, policymakers, and the public to ensure that CRISPR is used for the benefit of all humankind. As we venture further into the uncharted territory of gene editing, it is crucial to proceed with caution, humility, and a deep sense of responsibility. The future of CRISPR is bright, but it is up to us to harness its power wisely and ethically, shaping a future where this revolutionary technology benefits all of humanity.